Hi everybody, welcome back to Environmental Science Analysis with Dr. Lisa. Today we're going to talk about environmental time series analysis, and I use the word environmental here because it's important to distinguish environmental time series from all the other kinds of time series analysis that people do. Because, for example, if you look on YouTube and you look at time series analysis, you're going to find a lot of stuff that doesn't have anything to do with the environment. There's a lot of analysis of things like stock prices, which is great. But environmental time series analysis has some of its own unique problems. And so we're going to talk a little bit about the theory in this lecture, this very short video. And if, the actual process of doing it, I've already made videos of how to do that using some nonlinear curve fitting and even linear curve fitting in something like R. But we're going to talk a little bit about the theory now. So the question is, why? Why do we do this? Actually doing time series analysis is fraught with a lot of problems. And one of the things I want to really press home to you is how quickly the future becomes the past, right? So I was driving in the car the other day with my son, and the Prince song 1999 came on, and we're jamming, we're having a great time. And my son says, I was born in 1999. And I said, this song came out in the early 1980s when 1999 was the future. And now it's the past, and that happened so quickly. I feel like I blinked and my life went by. And people who do climate change models were making lots of predictions about what was going to happen to our climate in the year 2020. And guess what? It's 2020 now. So it's easy to see these time series analysis as this very intellectual exercise that has no bearing in reality. But in the real world, what we do here matters. And over the course of your career, very quickly, your predictions are going to be tested. So why do we fit our data to models? And here I'm really talking about models in general, not just time series models, but any models. Why do we do this? Well, I would argue that there's three main reasons. One is interpolation, right? And here's our little graph down here that's trying to describe the difference between interpolation and extrapolation. Interpolation is figuring out what's going on here where you do have some data, and you'd like to figure out what's going on in between the data points. So as an example, if you're trying to estimate cancer risk among people who live in New Brunswick, you might have a model that describes their daily activities. Maybe it's a 24-hour model, or maybe it's even shorter than that because they spend 8 hours at work and 8 hours asleep. So you might have a time step in your model that's less than 24 hours. But one of the sources of exposure to toxins might be atmospheric concentrations of PCBs. Well, we measured PCBs in the atmosphere of New Brunswick for many years, but we did it every 6th day. And so you might need to interpolate between those measurements to figure out the concentrations of the PCBs in the air every 8 hours. And that you would feed into your model. So we do that kind of interpolation all the time. That's quite common. Then the other reason that you might want to fit your data to a model is to extrapolate. And usually when we're talking about time series models, or we're talking about extrapolation, we're talking about forecasting. We're talking about thinking about the future, right? Doing a forecast to try to predict what's going to happen to PCB concentrations in the Hudson River over the next 20 or 30 years. But it is also possible sometimes to do an extrapolation into the past, and that's called a hindcast, and that's a real thing. We do sometimes need to do that. That can be important in environmental science too. Now, making forecasts is sort of the number one thing that people do with time series analysis, and it's also the number one most problematic thing you can do. And so I like this wonderful famous quote by Yogi Berra. Yogi Berra was a player for the New York Yankees and then later became coach and manager of the New York Yankees. And he was famous for saying things that were incredibly stupid and obvious and yet also really profound at the same time. And so this is a quote by Yogi Berra where it says, "It's tough to make predictions, especially about the future." And the most important thing that you can remember from this lecture is that making forecast predictions is extremely dangerous and very likely to take you wildly astray. But there's a third reason why we might fit our data to models, and that is to understand processes. So for example, when you look at the time series model video where I do the nonlinear curve fitting using PCB data from New Brunswick, one of the things we're doing is trying to see, is there a long-term decline in PCB concentrations? Is that time constant statistically significant? And, and sometimes the answer is yes, sometimes the answer is no. But that answer will tell us, is there truly a decline in the concentrations? We also, in that example, fit our data to temperature. Turns out temperature is an important driver of PCB concentrations in the atmosphere, and the fact that that fits with our model, and our model shows that that's statistically significant, helps us to understand something about the processes that are driving PCB concentrations in the atmosphere. So I would argue that because forecasting is so fraught with all kinds of problems, that understanding processes is actually the most important and most valid reason why we might fit our data to models. Okay, now, how do we do this? Okay, well, there's a couple of different ways. Very frequently we use linear models, right? And that's because they're easy. That's what people do. It's fine. In the example that is in the video on nonlinear curve fitting, I actually did some linearization where you take the natural log of concentration and 1 over the temperature in order to fit your data to a line. And that's fine. It has some so it's not actually a linear model. We're using log, we're using exponents, we're using 1 over temperature. So it's not truly a linear model, but you can use the linear equation to fit it and to come up with your coefficients. And that's fine, but I would argue that we're living in the 21st century. We have great computing power. There's no need to linearize data unless you really have to. We have the computing power to do lots of nonlinear curve fitting. So I would argue that there's no point in doing it if you can avoid it. So then the question becomes, okay, whether I'm doing a linear model, a nonlinear model, what is my criterion for deciding whether the model fits or not? And usually, a lot of the time, we're minimizing the sum of squared residuals. That's what we're doing when we're doing a linear regression or a multiple linear regression. You're minimizing the sum of squared residuals. Notice that there's no weighting. The data points all count equally. None of them are weighted any more than any others, okay? And so this is a very standard approach, and if you don't know how else to do your model, this is a good place to start. But when you linearize by doing things like taking the log, you are weighting your data. Whether you know it or care or not, you are putting some weight on more data points than others. And so that's an important thing to keep in mind, that if you're minimizing sum of squared residuals and you're not thinking at all about weighting your data points, you might be weighting them without knowing it, and you need to really think about whether that's what you want to do or not. As an example, it is possible to weight your data points, and an example of that we're going to do later in the semester is positive matrix factorization. The PMF software that we're going to use is PMF 5.0. It was developed by the US EPA, and it minimizes this thing called the Q function. It's not very important that you know what the Q function is, but all you have to do is understand that the Q function includes a weighting, and the way you weight the data in PMF is by giving the program an uncertainty matrix. So you're telling the program, here's my certainty or level of uncertainty about every single data point, and the Q function takes that into account so that you're spending more time and effort trying to fit the data points that have a low uncertainty and less time and effort trying to fit data points that have a high uncertainty. So it is certainly possible to weight your data by these functions. It makes everything more complicated, but it's totally doable. Now, there are plenty of other ways, other metrics that you can use for your fitting criterion. You can use root mean square error. You can use average absolute deviation. There's a lot of different things you can do, but it's up to you to come up with a reasonable criterion for what is your model fit and for deciding which model fits are better than others. Next question, how much data do we need to do what we want to do? And the short answer is more. You always need more. More, right? More. Okay? You never have enough data. This is very important to understand, particularly about environmental curve fitting and time series analysis, because you never have enough data. Most environmental processes are happening over the time scale of years, and collecting data over many years is a tough thing to do. It's tough to get data that is reliable and all measured exactly the same way and comparable across many years. It's just tough to do. Now, the longer answer is that you can use models to decide exactly how many samples and how often you need to sample to detect whatever your target is. So for the Upper Hudson River, they do it's part of a Superfund site, and all Superfund sites have to be assessed every 5 years. And so they put together a sampling campaign that would detect a 5% decline in PCB concentrations over 5 years. That sounds easy, right? Piece of cake. Turns out you need thousands and thousands of samples to be able to detect that change because there's so much natural variability in the system. But the point is that they did use a model to make a real informed decision about how many data points they were going to need. And then you always have to ask yourself, not just time series modeling, but any kind of modeling, is the data that you have good enough to answer the question that you are asking? And the short answer is no. That should always be your first assumption. The data you have is probably not going to do it. You know, you're always getting these sort of partial answers and hints, but there's always a need for more information. So you should always have that sort of bias of assuming that the data is not good enough. And then you really have to convince yourself and convince, you know, the readers of your publications that your data really is good enough to answer the question you were trying to ask. So the problem with environmental time series analysis is all the things that can go wrong. So there's this famous movie from way back in 1981 when I was just a youngster called Body Heat, and it's a film noir kind of thing about this guy here, played by William Hurt, who is talked into murdering his girlfriend's husband. And he goes to this guy, Teddy Lewis, who's played by Mickey Rourke, back when Mickey Rourke was like young and hot. And he gets this guy to help him build a bomb. And Mickey Rourke is asking him, like, seriously, I have a question for you. What the F do you think you're doing? This is not stuff that you should be messing with. Anytime you try a decent crime, you got 50 ways you're going to F it up. If you think of 25 of them, then you're a genius. And you ain't no genius. So I thought of 25 ways that you can go wrong every time you try to do a time series analysis because I am a genius. And so here's just some examples of all the things that can go wrong. Okay, so seasonality. Current use pesticides might get applied at a certain time of year, but that time of year might be shifting depending on climate change, depending on, you know, which crops they decide to grow this year. Maybe they rotate their crops and grow different things every 3 years. Maybe they used to apply this pesticide in the spring, but now they've changed to fall. You know, all kinds of things like that can go wrong, can be different, you know. Then there's meteorology, droughts, hurricanes. Hurricanes are notorious for messing up everybody's research. I went to graduate school in Baltimore, and every time a hurricane would come toward the Chesapeake Bay, all of my fellow graduate students would panic because they'd be like, everything that I've been studying is going to get completely blown out of the water by this hurricane. Climate change obviously is changing everything all the time. So you think you understand how your system is working, but then the climate changes, and guess what? You don't understand anymore. The location from which you take your samples is always problematic. They build a building next to where you like to take your samples, or they force you to move, or the power goes out, or, you know, things happen all the time. We've been sampling over at Rutgers Gardens for years, but there's always something going on there. They move things, they cut down trees, it changes the meteorology, everything is a problem. The power goes out. So these kinds of locations are, you know, if you're sampling CSOs, guess what? Your CSO's gone. They capped it off. It's not there anymore. So location is always a problem. Species. If you're, let's say you're trying to, you know, collect samples of a certain type of fish species. Well, you know, new species can get introduced, which totally changes the food web. You know, there are fishing seasons, and the fishing season might change. There might be bans on fishing. There could be, you know, any number of things could happen that could cause the whole ecosystem and the whole food web to change. It's very difficult to control the age and sex of the fish that you're catching, and to control their lipid content and how healthy they are. And there's a famous phrase, "Fishin' ain't catchin'." You know, you go out fishing hoping to catch a certain type of fish, a certain species, a certain size. But what you go out there hoping to catch isn't what you actually catch. You get what you get, and you don't get upset. Then there's all kinds of human activities that can mess things up. Bans. Certain chemicals could get banned. You spent your whole life studying, DDT, and suddenly Rachel Carson comes along and it's banned. And guess what? Everything's different now. They're constantly upgrading sewage treatment plants, capping off CSOs, things like that. They might change water management. There are places where, even because of lawsuits sometimes, the way that dams are managed and the amount of water that they let flow over them could change. There's all kinds of lawsuits about how much water gets allocated to which people, and depending on the outcome of those lawsuits, the hydrology of systems can change completely. In the New York, New Jersey harbor, they do dredging all the time, and a few years ago, I forget how many years ago, they actually did a big dredging project to deepen the harbor, and now that the harbor is much deeper, the hydrology is totally different. And then, of course, the big thing that happens to us in the environmental field is that the money runs out. And suddenly we don't, that's why we're not measuring PCBs in the air in New Brunswick anymore, because our money ran out back in 2011. And then there's all the black swan events, things like 9/11, things like dam failures. Dams just collapse for no reason. Wildfires, COVID-19, right? That's messed up everybody's attempts to do all kinds of time series analysis, because it's just something that came out of the blue and has changed everything. It's changed all kinds of environmental emissions. It's changed land use. It's changed how much people travel in their cars. It's changed everything that you can think of. So that's just a cautionary tale on all the things you got to think about before you do your time series analysis. So go ahead and do it by all means. Do it, fit your constants, make your predictions, but keep in mind all the things that can go wrong.